Medicinal plants have bioactive compounds called phytochemicals that are used to treat various human diseases. Phytochemicals have two categories, primary and secondary constituents. Primary constituents have compounds like chlorophyll and amino acids, while secondary constituents contain compounds like alkaloids.
In this video, we will focus on the secondary constituents. Some of the useful major groups of phytochemicals can be divided into several categories that include alkaloids, flavonoids, essential oils, lectins, polypeptides, phenolics, polyphenols, tannins, and terpenoids. But we will focus on alkaloids, phenolics, and flavonoids today. In general, alkaloids are a group of natural nitrogen-containing basic compounds with low molecular weight, synthesized from amino acids. It is used as an anesthetic agent which block the feeling of pain and sensation. A known example would be morphine extracted from opium poppy. Moreover, alkaloids has an anti-inflammatory activity whereby it inhibits or regulates important inflammation mediators like cyclooxygenase 2 or COX2.
Next is the simplest bioactive phytochemical, phenolics. Phenolics have a single substituted phenolic ring and are toxic to microorganisms due to the sites and numbers of hydroxyl groups on the phenol groups. Also, highly oxidized phenols possess more inhibitory action. So why is phenol toxic to microorganisms? This is due to enzyme inhibition by the oxidized compounds, possibly through reaction with sulfhydryl groups or through more non-specific interactions with proteins. Polyphenols can form heavy soluble complexes with proteins and may bind to bacterial adhesions. As a result, reducing the amount of surface area available on the bacteria to bind to a cell.
Lastly, flavonoids have a polyphenolic structure and are potent inhibitors for several enzymes like xanthine oxidase. Also, flavonoids are useful as an antimicrobial as they can form a complex with extracellular and soluble proteins, which then binds to the bacterial cell wall. More lipophilic flavonoids may also disrupt microbial membranes. Hydroxyl groups absent from flavonoids' beta rings have more activity against microorganisms as it will target microbes with OH groups on its membrane. As a result, flavonoids are widely used in pharmaceutical companies due to its antioxidative, anti-inflammatory, anti-mutagenic, anti-carcinogenic properties and their ability to modify key cellular enzyme functions. On that note, this is the end of drug discovery of natural plant products from plants. If you wish to know more, click on the following references below. Thank you very much for your kind attention.